just like any other skill that you're trying to develop across, just practicing it and trying to get incrementally better before test day is the best way to avoid some, bad outcomes.
Evan back for some more MCAT podcasts. How you doing, my friend?
Doing well. Ready to explore some bio-biochemistry.
Bio-biochem. Don't say those forbidden words in this household. Bio-biochem. So we finished cars in a normal full-length kind of testing environment, real testing environment. Bio-biochem comes after lunch. What are your thoughts for lunch breaks for students?
Lunch breaks, yeah. I think obviously you should eat something. I think that a big thing for lunch break, it's the longest break. You get 30 minutes. It can be good or bad. It can be very restful and allow you to come back in the biochem section with a lot of energy, kind of a second wind. But it could also be tempting to kind of overthink and go back and think, oh, if I had another 5 minutes, I could have gone back and done this or that or whatever. That's ultimately not going to be helpful. So we want to make sure that we're always forward thinking, right? Maybe you missed a question here or there that you wish you could have had back. That's okay. Make up for it in the next half of the test. But with lunch itself, I be really conservative with what you're eating. Eat something you've had several times before. Don't try out some new recipe on the day of the test and risk some GI problems.
Montezuma's revenge coming for you.
Yeah. Be conservative with that. I think for testing I had a PB&J.
Yep.
And a granola bar.
Yeah.
So, yeah, think something like that maybe.
Can't go wrong. Unless you're allergic to peanuts. Do not have PB&J. Awesome. All right. So we come back from lunch. You got to go do the whole check-in process. How do you deal with stress of going through that whole process to get back into the testing center and go back to your seat?
I tried to start checking in early. The worst thing that you can do for your stress levels going into there is to take an extra long lunch and then try to, shuffle through the line of people back in a testing center and cost yourself, even if it's just 30 seconds or a minute. That honestly is probably not going to have a big difference on your score, but that's going to make you super stressed walking in there knowing you've already lost a little bit of time. So don't risk it. It's okay to take a slightly shorter lunch. And then maybe you have a couple of minutes left in your lunch break where you're just sitting at the desk, where it's quiet. There's no one else rustling or nervously chatting, and you can kind of just have a second to yourself. That's kind of what I tried to do, was a few seconds of solace there before I went back into the storm.
Yeah, got it. Okay. Well, let's go ahead and jump into our passage again. This is bio-biochem, passage 1 from the Blueprint MCAT Diagnostic exam, which everyone gets for free over at blueprintmcat.com. So, let's ride this wave into fun. So we learned last week's episode, covering kind of that passage 5 of cars, reading, stopping, highlighting. Do you do any sort of difference in reading passages for the science sections versus cars?
Somewhat, a little bit different. I would say the cadence is pretty similar, where I like to kind of take stock of stuff paragraph by paragraph, but the focus of my highlighting is a little bit different because, like we talked about, in cars, there's absolutely no outside knowledge that's tested on. In the science sections and in bio-biochem, there's lots of outside science knowledge that can be tested in. So sometimes I like to change my highlighting a little bit. So I'm still trying to incorporate the key ideas. And, if there's an experimental style passage, I like to kind of try to include the results and the hypothesis and stuff like that could be helpful. Mm-hmm. But I also sometimes try to highlight any things that I know are MCAT testable, or make note of them, since, they're not going to ask me a question about something that's not required knowledge. But if they draw a connection between this new passage information and amino acid composition, or this passage information in enzyme kinetics, that is possible for them to test on. So it's really good to kind of focus on that type of stuff for these science passages.
Okay.
We'll just do this kind of the same way, I guess. So kind of just start reading paragraph by paragraph. I'll toss you some questions, Ryan, kind of see what you're thinking here.
Okay.
Paragraph 1. Cataract generation triggered by lens injury is considered to be driven by the action of the proliferation and epithelial mesenchymal transition of lens epithelial cells as a complication of injury and inflammation. The epithelium quickly expands, creating a 100-fold increase in the number of cells. In response to trauma, irritation, or surgery, cytokines and growth factors increase in the aqueous humor and stimulate LECs to proliferate and undergo EMT. This response is significantly diminished in organisms where EMT is inhibited by the inactivation of transforming growth factor beta, TGF-beta, especially TGF-beta 2, the major isoform in the aqueous humor. There's a lot of jargon there.
Just a little bit.
Just a little bit. And a lot of it is honestly, I would say, more into the weeds than the MCAT is usually going to be worried about asking us about. We don't need to have a deep anatomical or physiological understanding of what lens epithelial cells do.
Mm-hmm.
Presumably, I don't know, you probably learned that in medical school, Ryan. You probably, maybe eons ago, saw this, right? But
I'm not that old. Come on.
Yeah, 5 years ago when you were back in medical school.
Yes, just 5.
Just 5. Quick 5. But there's some stuff here that is probably I don't know. Is there any stuff here, Ryan, that you see that either seems like it's hitting on stuff that might be testable, that's some of that more foundational level biology, or any stuff that just is stands out to you as being important because it's kind of summarizing everything in this first paragraph?
Yeah, I mean the last sentence kind of summarizes a big part of it. The beginning part is the what happens, right? What's causing this kind of this cataract generation and the definition of that, of what's going on here. And then the big part here that I think we focus on is this part here that focuses in on, hey, there's this inactivation of this TGF beta and specifically beta 2 that's going to help.
Yeah, exactly. I like that a lot. I love how you pointed out in that first sentence, we're getting kind of a description. They're saying there's this thing that happens, cataract generation, and we know that we are suspecting what's causing it, right? Because they're telling us this is driven by the action of ... That type of language is giving us a causal relationship. It seems like the cause is this proliferation and EMT of the lens epithelial cells, and the cataract generation is the effect of that cause. So that's something they like to ask us about, or potentially something that will be introduced that they're trying to study maybe as part of a cause and effect relationship in an experiment. Great thing to keep in mind, though. Perfect. Moving on to our next paragraph. During the process of EMT, LECs undergo cytoskeletal rearrangement with the addition of a large amount of extracellular matrix proteins such as collagen and fibronectin. In anterior cataracts, the proliferation and EMT of LECs in situ leads to the formation of opaque plaques just beneath the lens anterior capsule. This one's got a lot more jargon here, but is there any stuff here, Ryan, that you see reminds you of stuff from your intro to bio class, or just that foundational stuff that does seem testable here?
Extracellular matrix proteins, I think, is a big one here. And then maybe this in situ formation of plaques. I don't know.
Yeah, I mean, honestly, I think that, probably it's a good idea to be a little bit familiar with this idea of extracellular matrix and, the cytoskeleton. Those are good things to have at least a foundational level understanding of, not necessarily a super deep understanding. And then I really like that second highlight you made. I don't necessarily think that we need to know what an opaque plaque is, but I like that it's telling us this leads to something else. So it's giving us another one of those cause and effect relationships, which can be really important to keep track of here.
Yeah.
Okay. Paragraph 3 now. Paragraph 3. The production of TGF-beta is catalyzed by histone deacetylase 1, or HDAC1. HDAC1 removes acetyl groups from N-acetyl lysine groups on histone. Once synthesized, TGF-beta is secreted by activated monocytes in the aqueous humor. Since monocytes mediate almost 80% of the local TGF-beta, scientists tested the theory that TGF-beta is responsible for the monocyte's impact on LEC production. The number of LECs expressing MIB1, a nuclear protein, was utilized as a measure of LEC proliferation and expected EMT. That's dense. That's dense. There's like an unbelievable amount of alphabet soup in there, but there's a couple things that we can maybe like keep track of in here. And I think going back to those basic things, can we find any cause and effect relationships or any hypotheses that they're making here?
Mm-hmm.
Brian, do you see any stuff like that? Any language that's indicating to us what they think is happening?
Yeah, so this monocytes mediate the local TGF-beta, responsible for impact on LEC production, and then this MIB1 as a measure of LEC proliferation and blah, blah, blah.
I like all that stuff here. So honestly, that middle sentence there that you pulled out, I like a lot, because that honestly sounds to me quite a bit like a hypothesis. It's like, we know something. There are these monocytes that do, a lot of that mediate a lot of this local TGF-beta. In light of that, the scientists are testing some theory. That sounds like the beginning of an experiment, and that's good to keep in mind because it's telling us what are we interested in. It's honestly giving us kind of a preview to the variables that are probably going to be used in whatever experiment and testing we're doing, which would be a good kind of insight there.
Mm-hmm.
Then we get our experiments here. So just a label here says experiment 1, and then I'll give us a little bit of our description here. So in experiment 1, after simulated injury, monocyte/LEC cultures were treated with an antibody that lyses all but a negligible fraction of available monocytes. A division of these cultures was also treated with a TGF-beta agonist. A second set of cell cultures was treated with antagonists of epithelial TGF-beta receptors, TGF-BR1 and TGF-BR2, as shown in Table 1. Okay. So that's a little bit of a description, and I really like this description because it's telling us in words what we're seeing in the table. And I think for lots of students, kind of dissecting these tables, these figures can be really difficult here. I don't know. That was kind of, for me, one of the biggest things that I had to get over the hump with in the bio-biochem passage. How do you feel, Ryan? Do you love tables? Do you have a long story relationship with tables and figures and stuff, or you kind of...
I think it highly, highly depends on what's in there. I'm not for or against. I just hope I understand it.
Yeah, that's a good. I will kind of preface this with, in my opinion, when we're going through a science passage, you can get sometimes passages that have 2, 3, 4 different tables or figures. And sometimes you won't get any questions about some of the tables or figures. For that reason, I usually only, on my first pass through the passage, maybe spend 5 or 10 seconds glancing at each table or figure. So that way I'm not, dedicating time to a figure that might ultimately not get me any extra points.
Yep.
But if it gets referenced, or if I know I need to use it to understand the results, then maybe we can dig into a little more. So looking here at Table 1 and looking at what they told us from this little preamble or this description of Table 1, it seems like we're kind of trying to test here what's happening when we have either the antibody present, that's an anti-monocyte antibody when it's present or it's not present. And then we're also looking to see what happens when we have the agonist present or when we have the antagonist present or absent. So we're kind of saying what's happening when we have these things or don't have these things. And there's 4 different things that we're interested in and kind of saying, is it here, is it not here? And that's kind of the mix of different results that we're reading.
Mm-hmm.
Am I super concerned with the results right now? Probably not, because I know, based on just that 10-second glance over this table, I have a good enough understanding of what's going on, what's being tested, that I can identify if this is being asked about in a question, and then know I can come back to it to overanalyze it later.
Okay.
So we'll maybe come back and worry about it later, depending on whether or not we see a question. But in the meantime,
I pray we can ignore it.
That's what I say too. Whenever I see a really difficult like table with all these things going on, all these little side lead and stuff, I'm like, please don't let there be a question. Yeah, we'll see. Sometimes there is, sometimes there isn't. But regardless, we don't want to spend too much time digging into it until we know we have to.
Yep.
All right. Then we're introduced to experiment 2. In an effort to determine whether MIB1 is associated with LEC regulation, LEC density was assessed during the course of epithelial growth in wild type and MIB1 null cell cultures as shown in figure 1. Okay, so Ryan, based on that description here, and based on what we can see as the figure, which the audio listeners might not be able to see, but kind of tell me, what does it seem like the variables are here? I think that's a good thing to glance at in our 10-second look over of this graph. What are we changing? What's the independent variable? What are we changing as the researchers?
The MIB1 association here.
Yes, yeah, whether it's a wild type or whether it's a null MIB, whether they don't have any of this MIB1. Mm-hmm. Awesome. Then what are we measuring? Right, what is the change we expect to see or that we're measuring to see if there's a change as a result of that independent variable manipulation?
We're looking at the LEC density.
Perfect, great. I think that is a great understanding. Honestly, that's like maybe even more than I would even expect us to get on that first pass through. That is really, really good. Identifying those variables, because that's giving us a very good scope of what's going on here. Okay, dense passage with lots of like alphabet soup. Really, that's what I always call it whenever I see anytime there's genes or proteins and stuff like that. The naming conventions are a headache, so we want to be really careful making sure we're picking the right ones.
Yeah. Oh, all right. Now I'm worried. Question 1. The catalyst for TGF-beta production most likely plays a direct role in A, gene silencing, B, ribosomal inactivation, C, transfer of an acetyl group from acetyl-CoA, or D, gene activation. The catalyst for TGF production. I don't know. So, this TGF production, or we were first introduced to TGF here in this first paragraph. Mm-hmm. I have no idea. Oh, wait. So here. Production of TGF-beta is catalyzed by histone deacetylase 1. HDAC1 removes acetyl group from N-acetyl-lysine groups on histone. And then TGF is secreted by activated monocytes in the aqueous humor. All right. So basically, what is this? Histone deacetylase 1. So transfer of acetyl group from acetyl-CoA. So deacetylase, acetyl group. Deacetylase, acetyl group. I'm going to go with C and move on.
That's a very tempting answer.
That's because I have no other idea.
Yeah, so this is, you identified, I liked everything up to that point. You read the question, you said, okay, I need to figure out what is the catalyst for TGF-beta production. You went through, you scanned it, you found it. It's this HDAC1, this histone deacetylase 1. And all they tell us about it, point 1, it helps with the catalysis of the production of TGF-beta. And the second thing they tell us is that it's doing some of these chemical modifications to histones. So, Ryan, do you remember what a histone is or what a histone does?
Has to do with genes.
It does. Yeah, so histones are these protein complexes. If you think about it, the way that the DNA is wrapped up when it's in its condensed form in its chromosomes, it's like these beads. And then around that bead, it's like the DNA is spun around it tightly to make it really condensed and easily packaged in, a small place inside the nucleus. So what the histones are really important for is DNA package. And there are modifications on these histones that we should be familiar with for the MCAT. This is where a little bit of that outside knowledge does come into play here. And really the only outside knowledge we need to know here is that histones have to do with DNA and like whether DNA gets turned on and off based on whether the DNA is attracted to the histone or repelled from the histone. Because if it's closely and it's attached to the histone, right, it's stuck and it can't move and it can't get transcribed. Whereas if it's repelled from the histone and there's some separation, we have room to get the transcription machinery there. So here, what happens when we are deacetylating a lysine group? So lysine is one of our amino acids that we want to be familiar with. When it is acetylated, lysine is neutrally charged. It doesn't have any charge at all. When we deacetylate lysine, it gets a positive charge. DNA is negatively charged always. That's just what DNA is. So the negatively charged DNA, once that lysine gets deacetylated, they come together, right? It's positive and negative, opposites attract, and they get stuck together. So after we deacetylate that lysine group, we're causing this DNA, this gene, to get stuck to the histone, which makes it inaccessible to our transcription machinery, to our RNA polymerase. That ends up causing this gene silencing, right? Because if the RNA polymerase can't get there to that gene anymore, we're going to have no way to transcribe it, and therefore it's going to turn off transcription. So kind of a takeaway here that I get here from Ryan. You did an absolutely perfect job on the passage part of it, right? You found the right information in the passage and you got it. This is showing kind of how the cars is a little bit different though, because in cars, that's all you got to do. It's only look at the passage. In the science sections and the science passages like bio-biochemistry, we have to marry the two. We have to marry our outside knowledge from studying, the biochemistry, the biology, and we have to put that together with the understanding of the passage.
So what's the answer here?
A. Oh, so sorry. Yes, yeah.
It's your choice A. Gene silencing.
Gene silencing, because, like I said, once we deacetylate that histone, once we make it go from neutrally charged to positively charged, it causes the histone and the DNA to get stuck together. That silences the DNA, the genes in the DNA, because that removes any space around the DNA to let the transcription machinery come in and do its transcription.
Got it. Yeah. That's hard.
Yeah, and that is a tough one. It's going to require some studying for sure. But, if you were a student of mine, I'd be like, okay, cool. What I noticed from there is that you did a really good job with the reading of the passage and comprehension of the passage. And it was just a little bit more of that, outside content knowledge.
Yeah.
Cool. Throw that in a flashcard, study it a few times, go watch a video on it. There's plenty of information online. Make sure that you feel good about it. The next time you see a question about histones, you're going to know.
Yeah. Got it. All right. Question 2.
Question 2. Monocytes, in conjunction with epithelium-derived factors, can act to facilitate which biological process? And our 4 answer options are A, fatty acid oxidation, B, transfection, C, lipid synthesis, and D, host-immune response. All right. There's kind of one key word or phrase in the question stem here it's a monocyte. I think that's the key to understanding this question. What is a monocyte? It's kind of
they're white blood cells. A typo.
They're white blood cells.
Yeah. So white blood cells is immune stuff. So that's what I would go.
That's it. That's it. That is it. This actually has very little to do with the passage. You don't need. You could have just had this question without the passage and still known the right answer. This is one that we kind of call a pseudo-discrete question because it's part of the passage, but it acts exactly like a discrete question because you can answer it without the passage.
Yeah.
Awesome. Cool.
All right.
Moving. That's right.
Whew. All right, question 3. After injury-induced cataract formation has begun, Which of the following are least likely to be found in nearby monocytes? So again, least likely. A, TGF-beta transporters, B, TGF-beta receptors, C, tight junctions, or C, cytokines. So if we go back up to super dense, what's going on in this world during the process of this epithelial mesenchymal transition, so where this injury comes from, we have lots of stuff going on. So we have monocytes, which are doing TGF beta stuff, transporters, receptors, okay. Cytokines, we know, going on, some bad stuff, cytokines. Tight junctions are this weird, like, which one of these are not like the other? Like, there's, like, stuff going on, and then tight junctions to me is like healthy cells. They're sticking together. Oh, wait, these are not healthy cells. They're injured and pissed off and leaky and all kinds of stuff. So I'm just going to see, like, ignoring everything else.
I mean, that's not a bad try. If you have that, which one of these is not like the others, because this is, a multiple choice test, 3 of the answers have to be wrong, 1 of them has to be right. If you notice 3 of them are very similar to each other, that can be a good hint. I'm not going to say that's an automatic, it's always right, the one that's different than the others. But in the absence of like a better idea or better guess, that's not a bad rule of thumb there. Brian, you're right. That is a good way to do it. Another thing that we can think of here is you mentioned correctly that monocytes are white blood cells. So white blood cells are important for immune system. Are white blood cells, are they stuck in one place? Are they moving around? How do they kind of act?
How do white blood cells act?
Yeah.
I don't know. They go to a source of injury, infection, whatever, and clump up and make pus.
Yeah, they're zipping around the circulatory system, around our blood and the lymph system and different stuff like that, right? They're moving around though until they find some area to help deal with infection. So if we're talking about just normal monocytes, right, these white blood cells, they're probably unlikely to have tight junctions because tight junctions make them anchored down in place so they can't move and they're stuck. Mm-hmm. That's the opposite of the function of the monocytes, which we need to be able to move around, go to different places to go and find that infection and ward off any invading bacteria or viruses. So that's another reason why C is the least likely thing, therefore the right answer.
Yeah. Could you read into this? I get super nitpicky with these types of questions, right? Tight junctions, are tight junctions really found in cells or on the surface of cells? So it's like, I don't know.
I mean, that's a good point. They're a part of the tight junction is going to be in a cell because, it has to have some anchoring point inside the cell, and then it allows them to have, the anchor point from one to another. Is it in the cell? Ish. But it's certainly a better answer, right? Again, going back to this, it doesn't have to be a perfect answer as long as it's significantly better than each of the other answers, which tight junctions is significantly better than these other answers since they are all explicitly mentioned as being involved with the monocytes action in this cataract generation.
Yeah. Got it. Okay.
Perfect.
Whew. All right. So what, two for 3 so far?
Yes, 2 for 3. And moving on to question 4 here. Tell us. The structure of lysine is shown below. And we see a structure of lysine up here, a little diagram of lysine. And then the question is just, this molecule is best described as A, aromatic, B, polar and basic, C, nonpolar and not aromatic, D, hydrophobic.
So this is the time to plug MCATflashcards.com so you can get access to 1,600 flashcards so you know all of your amino acids inside and out.
Yeah, knowing the amino acids, very important. Although I will quibble here a little bit. It is incredibly important to know your amino acids. You should. That is a worthwhile part of your time. That's one of the most like highest yield guaranteed things you're going to get questions about,
Yep.
we actually can answer this one with only foundational knowledge of general and organic chemistry with no knowledge of the amino acids. Though, so it does show us the structure.
Yeah. So aromatic to me equals ring.
That is one of the criteria. Yes.
Yeah. So to me, it's not aromatic because there's no ring there. At least that I can see in this diagram. Maybe there's some funky thing I can't see. So I'm going to get rid of answer choice A. Hydrophobic, I forget what structures make something hydrophobic. I forget if alcohol is hydrophobic or not, because there's an OH on there, right? Or NH2. I don't know. Aren't the longer chain ones more hydrophobic? Or yes. No? Yeah. Okay.
So long chains ones are hydrophobic.
Yeah. So this is not short. I mean, it's got a little bit of a chain there, so maybe it's hydrophobic. I don't know, but a random piece of knowledge that's stuck in my head. So I'm gonna go, okay, maybe it's hydrophobic. So really, I'm stuck with B or C with my current line of thinking, polar and basic or non-polar and not aromatic. Well, we know it's not aromatic because we already got rid of answer choice A. There's no ring there. And so C is tempting, but it could be polar and basic, and it doesn't mention anything about aromatic. And so I don't know.
This is an interesting one. So let's go for, because I see two opposites, right? B and C is saying, one saying polar, one saying non-polar.
Yep.
So polar molecule is one where you have a lot of electrons in one part of the molecule and not a lot of electrons in another part. So there's a difference in that electron distribution. So are there some atoms that get a lot of electrons, some that get very few? Versus nonpolar, they're very evenly spread out.
Okay.
What do you think, Ryan? Even if you're just gonna, either you're confident or just a guess.
Well, I mean, to me, when you say that, well, I see two O's and an N on one side and only one N on another, and so that's not evenly distributed.
I agree. It's not evenly distributed, which means that it's true that it's polar. Therefore, we can definitely rule out C, non-polar.
Okay.
Yeah.
And so, answer choice B?
Answer choice B is correct.
Okay.
And this is, so therefore D is also incorrect. That's one we didn't really rule out earlier. We kind of hesitated on.
Yeah.
This is not. So hydrophobic just means it doesn't like water.
Yeah.
You're right that hydrophobic things have really long chains, but it has to be a really certain type of chain. It has to be a chain that's made up of carbons and hydrogens. Since carbon and hydrogen are very non-polar, lots of times non-polar and hydrophobic are kind of synonyms for each other. This thing has a lot of polar groups which are not hydrophobic, which is why this is ends up being answer choice B is correct. It's polar. It's basic because that those amine groups, those NH2s, when you put it in water, those take up an extra hydrogen and they have a positive charge, which makes them basic.
Okay.
So you can reason through it that way, or if you'd been, putting in work on your amino acid flashcards, this would probably take you 3 or 4 seconds. So different ways to answer it.
All right. Question 5. Given the findings in the passage, which additional result most strongly indicates that the TGF beta receptor 1, is that what the R stands for? Or TGF beta receptor 2 receptors regulates TGF beta dependent epithelial proliferation. Wow. So which additional result? So they're going to give us the answers are going to be additional results to the findings. Indicate that these TGF beta BR1 and BR2 receptors regulate TGF dependent epithelial proliferation. Okay, I'm already lost in the question. Answer choice A, treatment with TGF BR1 receptor agonist resulted in reduced MIB1 detection. Expression of TGF BR1 and TGF BR2 was observed to increase during intervals of heightened epithelial cell proliferation. Answer choice C, genes for several different receptor subtypes were found in untreated epithelium. Or D, treatment with the new TGF-br3 receptor antagonist decreased the number of MIB1 positive cells.
Yeah.
Wow. And I look at my clock and it says I'm running out of time, and I just pick one and move on.
Well, in the grand scheme of things, not a bad strategy. If you look at a question and you recognize that this is one that's going to be a time sink, and even after a time sink, it's going to be somewhat of a guess, it's good to hedge your bets, right? You don't want to sink in 5-6 minutes on one question that you're maybe going to get right, maybe you're going to get wrong. That's going to take away from easier questions later on. That being said, ideally, we answer every question with some level of confidence here. So one thing that I really, really liked that you did with our cars questions, Ryan, is that you rephrased them. I think rephrasing this question and kind of trying to think of what it's really asking for will be helpful and giving us a starting point here.
Be my guest.
Yeah. So let's kind of dissect this a little bit. So it's asking us which additional result. So they're going to provide a result for us as the answer choice. What result would most strongly indicate that these two receptors, TGF-br1, TGF-br2, regulate TGF-beta dependent epithelial proliferation? Okay. So if there was a result that's supporting something, that's strongly indicating something, we would probably want it to say, when I change the receptors, when the receptors are there, the proliferation happens. When the receptors are gone, no proliferation happens, right? That would show me that it's dependent on it, that this is, it's regulating it, because with them there, it's happening. With them gone, it's not happening. That seems like a pretty strong, relationship there, causal relationship. My guess is that something like that would be really good evidence here. So I made a prediction based on that rephrase about what type of answer choice I might be looking for. Right, did any of those kind of match that prediction from what we see?
I have no idea. This one scares me.
Okay, so with this one, right, we just read through those very complicated answer choices here. So A is just saying we treatment with TGF-br1 receptor agonist resulted in MIB1 detection. Okay. We actually never mention the agonist, the receptor agonist in the question stem, and this never makes any mention of the receptors or proliferation. So A is just kind of like ruled out because it's not even answering the right question. It's telling us some result that wouldn't support or contradict this kind of description because it doesn't even have the right actors, right? It's got the wrong suspects, so to speak. So A is wrong just because it's mentioning the wrong things. It's not even talking about TGF-br1, TGF-br2, and this proliferation. But I'd eliminate A for that reason. B, at first look, right, just in a second, I'm looking, oh, it has the right stuff. It's got TGF-br1 and TGF-br2, and it's got proliferation. Although just using actually just that lens, B is the only one that even attempts to answer the question by using the right, like, 3 pieces of information. The 3 things they tell us are TGF-br1, TGF-br2, and proliferation. It's asking us, what evidence do we need to say those 3 things interact? We probably want something that's making some type of comparison between the 3 of them, and B is the only one that actually does that for us.
This is why I hate the MCAT.
It's tricky, right? They will make the question so intimidating sounding, and then it'll be, some simple, relatively straightforward reason like this, where you can just say, oh, only one of them even like pertains to the question, and therefore I can be above the other. So
it's like the question is like, what colors would you observe if you mixed red and blue? It's like answer choice A, B, and C are just 3 randomly just weird names, and you're like, it's got to be one of those. And the answer choice D is like, it's going to be a mix of red and blue. And you're like, oh, that's the only one that has both of them. Oh my gosh, it's so frustrating.
It can be frustrating, but again, I like to think about this from the perspective of this is just practice, right? Whether this is a diagnostic test, whether you're just doing a practice exam, whether you're just doing your daily practice questions, whatever it is, it doesn't matter. On any given day, whether you get 3 out of 5, 4 out of 5, 5 out of 5, whatever, on that practice passage, what matters is that you're learning from any mistakes you make. So that way, tomorrow and the next day and the next day, all the way up to test day, you're making progress to the point where you're doing the best you can. Maybe we miss a question, but hopefully we learn something there.
We missed more than one question this time. We finished off cars strong the last few weeks, feeling confident at the end of the passage. This passage, I feel beaten down. I have peanut butter and jelly sitting in my belly because this is my first passage post-lunch. I feel deflated in terms of my knowledge and my ability to do well on this. I'm ready to go. I'm hitting void. I'm going to void this. I'm not even going to do it. Like, what do you tell a student right now?
That's a big instinct, I feel like, for a lot of people. We call it catastrophizing. That's like, or the other term that I like, it's chicken littling, right? It's like, oh, one thing happens, the sky is falling, you're running around, right? That is, I know it's easy for me to say this as someone who is looking back on this and has already done it. I can sympathize, though, understanding that, feeling very frustrated and disappointed when things don't go perfect, or when you see a really difficult passage, a really difficult question. Ultimately, that's okay. But for real test day, right, you want to get the best score possible, obviously. And chicken littling, catastrophizing is not going to get us there. The way that we avoid these things is by putting ourselves in that position, right? Making it so that we are used to doing this practice and very comfortable doing this. So that way, right, if you go before test day and do 10 practice tests before test day, I guarantee you will have come across passages in the past that are just as difficult, if not more difficult than this one. And every time you do that, every time you see a passage like that, when you're practicing, not on test day, but before then, you want to practice that skill of not chicken littling, of not catastrophizing, and making sure that you make sure you stay focused. And you mess up on your first full-length practice test, and you say, hey, you know what? I did chicken little this time. Second test, try to make it a little better. Third test, fourth test, etc. Just like any other skill that you're trying to develop across, just practicing it and trying to get incrementally better before test day is the best way to avoid some, bad outcome on.